Well, hello, everybody. This is Nilo Azad, and I'm so excited today to be able to welcome Dr. Maeve Lowery to our seminar series. Dr. Lowery has been a leading researcher in our field for many years. She initially completed her training in Ireland at the University College of Dublin, and after spending a few more years there doing subspecialty training, she came over to New York, where she took up a fellowship position at Memorial Sloan Kettering. She stayed there subsequently as faculty and quickly became known as a national and international leader in the care and development of new drugs for patients with cholangiocarcinoma. But a few years ago, the call of home brought her back to Ireland. And so she has continued to lead international efforts in developing new drugs for our cancer patients, and in particular has been very involved in leading the charge when it comes to IDH1 targeted therapy for our patients. And so I'm very happy for us to be able to hear from her experience, both in terms of what has happened in the last few years for IDH1 mutated patients, as well as what might be coming up in the future. So welcome and thank you.
Thanks so much, Nilo, and I'm delighted to be giving the talk today, and thank you so much for the invitation. Lots of old friends and old colleagues that it's lovely to see people all again. So first a bit of geography. I'm based in Dublin, Ireland. I was going to put a little map on. So this is a picture of the university I moved back to. It's the oldest university in Ireland, Trinity College, Dublin. And I hold a position there as Professor of Translational Cancer Medicine, but I also still practice medical oncology at our Cancer Institute. So Dublin is the capital of Ireland. Ireland is a country the size of the UK. We're not part of the UK, we're part of the European Union. And we're a country small of just about 5 million people in the Republic of Ireland. So that's where I'm broadcasting from today. And it's lovely to be able to speak to such an international group. These are my disclosures. So taking a little step back, how I thought I'd approach things today is to speak just broadly initially about IDH mutations in cancer, what an IDH1 mutation is and what it does and where we find it. More specifically about IDH1 mutations in cholangiocarcinoma and the genotype-phenotype correlations associated with IDH1 mutant cholangiocarcinoma. And then an area that's been really encouraging to see some positive data coming is the experience so far over the last really just 5 years with inhibition of mutant IDH1 in terms of phase 1 and then leaping straight to phase 3 drug development and the success there, but also the challenges and the lessons that we've learned from these early and later phase studies of IDH1 inhibition, and to illustrate somewhat some of the key questions that as translational and clinical researchers that we need to address over the coming 5 years. And then just briefly to talk about the new therapeutic strategies, specifically for IDH1 mutated cholangiocarcinoma that are currently in development in clinical trials, early and late phase. So to basics, IDH stands for isocitrate dehydrogenase. There are 5 genes coding for 3 IDH enzymes, and IDH1 and 2 are very similar in function and they share 70% of sequence. IDH1 is primarily cytosolic and IDH2 mitochondrial. And in normal physiologic conditions, they are essential components of the tricarboxylic acid TCA cycle. So IDH1 and 2 enzymes catalyze interconversion of isocitrate and alpha ketoglutarate. So I'm not sure that you can see my pointer right here. So, so this is a key area that IDH1 under normal physiologic conditions functions within. IDH2 mutations, somatic mutations, are typically found in catalytically active sites. And mutant IDH1 or IDH2 also actually alters the fundamental enzymatic activity of IDH1 and of IDH2. So it actually reduces alpha-ketoglutarate to the oncometabolite 2-hydroxyglutarate, 2-HG. This is unusual in cancer, that we have here a gain of function alteration, also called neomorphic, where the acquired genetic alteration actually completely alters the function of the enzyme. And this has important effects on the cell pathways and on some key signaling pathways within the cell. The accumulation of the oncometabolite 2-HG results in competitive inhibition of alpha-ketoglutarate dependent enzymes. And this results in dysregulation of multiple pathways, in particular epigenetic alterations, causing impaired cellular differentiation, and also crucially metabolic dysregulation. So here are just a schematic showing how the gain of function mutations lead to accumulation of 2-hydroxyglutarate. And at the same time, mutant IDH1 can create a heterodimer with wild-type IDH1. And that leads to further decrease in alpha-ketoglutarate, increase HIF1 alpha, and increase also in angiogenesis, further promoting the malignant phenotype. And so the accumulation of 2-HG promotes carcinogenesis through disruption of multiple key pathways that are central to cell regulation. So cellular metabolism, epigenetic regulation, effects on redox states, and also effects on DNA repair. And all of this creates opportunities in the therapeutic sense for therapeutic targeting specifically of IDH1 mutant cholangiocarcinoma or mutant cancers as particular subsets or genomically selected populations of patients whereby there may be the option for a particular precision oncology approach. So specifically in biliary tract cancers, IDH1 mutations are found in about 15 to 20% of intrahepatic cholangiocarcinomas, and much less commonly found in extrahepatic cholangiocarcinomas or distal cholangiocarcinomas. And they're really not found commonly at all in cholangiocarcinomas that occur in association with hepatitis or with liver fluke. IDH1 R132C and R132G are the most common mutations that are found. They're slightly different and more frequent than those found in other cancers where IDH1 mutations are prevalent, such as gliomas or chondrosarcomas or in AML. You can see here that there is also some anatomic distribution and patterns, if you like, of co-mutation and mutual exclusivity in terms of identification of IDH1 mutations. So they infrequently occur with FGFR2 gene fusions, for example, but can more frequently co-occur with BAP1 mutations. They can co-occur with KRAS mutations, but not frequently. And that probably also speaks to the more frequent pattern of identification of IDH1 mutations in the intrahepatic cholangiocarcinomas, as opposed to perihilar or distal cholangiocarcinomas. This is just an oncoplot demonstrating the pattern of commutation and mutual exclusivity. And you can see here highlighted in the red bar are the IDH1 mutant cholangiocarcinomas. Of course, here every vertical column here is a patient. And you can see with these sort of common genomic drivers in cholangiocarcinoma that you do see this pattern emerging of commutation and mutual exclusivity, in particular the co-location of BAP mutations with IDH1, the KRAS mutations, more frequently in IDH1 wild-type tumors, usually no co-occurrence of IDH2 and IDH1 mutations for obvious reasons. And so this pattern across the biliary tree, and in particular looking at sort of clinical applications or therapeutic targeting, that we don't commonly see the co-occurrence of the other obviously targetable alteration in cholangiocarcinoma, the FGFR2 gene fusions. We very infrequently see those co-occurring with IDH1 mutations. So accounting for somewhere around about the up to 20% of intrahepatic cholangiocarcinoma. So a substantial minority of patients with a rare tumor type. This just shows my paper a couple of years ago that differential patterns of somatic alterations between fluke positive and fluke negative cholangiocarcinomas. So here, this group here under the brown column are the liver fluke positive cases, and the pink columns, if you like, are the fluke negative cases. And when you look down here to the IDH1 mutant cases, you can see much more prevalence of IDH1 mutations in the liver fluke negative population. And so just speaking to that difference in terms of the patient population in whom you're most likely to identify the IDH1 mutations, not that they can never occur in fluke positive cases, but that they're much, much less likely to do so. And the same also true of IDH2 mutations, which occur much less frequently in cholangiocarcinoma, usually less than 5%. So there does appear to be a sort of a classic phenotype or molecular type associated with IDH1 mutant cholangiocarcinomas. And this is a lovely paper from 5 years ago, TCGA analysis of TCGA data looking at a combination of both somatic mutations, RNA expression, copy number alterations, and DNA methylations in this TCGA cohort. And this analysis clearly identified an IDH mutant enriched subtype, which you can sort of see here at the red box around it. And this particular cluster was characterized by low expression of chromatin modifiers, elevated expression of mitochondrial genes, and increased mitochondrial DNA copy number. So clearly demonstrating that this is a clinically relevant subgroup of patients with cholangiocarcinoma, and that the presence of IDH1 mutation does appear to delineate a particular subtype with clear characteristics and phenotype potentially that would lend itself to more personalized therapy. So IDH1 inhibitors, well, this is an area where thankfully we have some good news to convey, and I'll sort of walk you through the story of how this has emerged. Really, I think a success story, not just for cholangiocarcinoma, but for patients with rare cancers everywhere, because thanks in no small part to the advocacy of organizations like the Cholangiocarcinoma Foundation, patient advocacy groups, it really has discarded the narrative that we can't do good studies in rare patient populations. And so I think through a collaborative effort between industry, academia, international collaboration, really demonstrating that with determination that that this really can be done. So the first approved IDH1 inhibitor is ivosidenib, and this was initially approved in the first-line setting for treatment of adult patients with IDH1 mutant acute myeloid leukemia. And in those patients, it actually seemed to create what we call an epigenetic rewiring, whereby there was actually a differentiation effect within those patients that led to a substantial survival benefit and long-term durable control in many patients. So the initial study of ivosidenib in solid tumors was a dose escalation study, a single agent, and there was several cohorts of patients, glioma, where this most commonly identified, chondrosarcoma, about 50% of patients with chondrosarcoma have IDH1 mutations, and cholangiocarcinoma, which eventually went on to enroll almost 70 patients in a fairly large phase 1 study. So this is the schema for the study design. It was a phase 1 study, and in fact, there was very little toxicity identified in patients from the agent. And so the dose expansion phase, the dose selected for that is really based on pharmacokinetic analysis and rather than determination of a maximum tolerated dose. So it was a very well tolerated agent in the phase 1 setting. And in the cholangiocarcinoma specific cohort, it enrolled patients who had advanced disease, not eligible for curative therapy, and who had progressed following a gemcitabine-based regimen, and they just had to have measurable disease and good performance status. And so a lot of these patients were very heavily pretreated. Of course, one can imagine in a trial like this is a selection bias, because patients with advanced biliary tract cancers who are good candidates suitable for the intensity of a phase 1 study typically may be those patients who are fortunate to have a more favorable disease biology. Perhaps they've responded well to treatment in the past, perhaps a slightly lower volume of disease, So of course we know we're selecting out those patients. And at this time when the study was begun, it was not routinely available to all patients to have molecular profiling performed. And I know that now most patients with new diagnosis of cholangiocarcinoma will know that it is essential to identify the molecular subtype. But back then it really wasn't quite as commonly available to people. And so the patients who had molecular profiling performed were those patients who had done well enough with their initial lines of treatment to actually get the testing done. So a very select patient population, but still relevant. So the initial results of the phase 1 study published a couple of years ago, the initial median progression-free survival, 3.8 months. Well, you know, that wasn't as dramatic as we might have hoped for. So you see median progression, 3.8 months. Well, is that better than what we might expect with chemotherapy? Is it better than what we might expect with observation in patients who perhaps have a better disease biology? Are we seeing an effect of the agent at all? And certainly what we're not seeing is a very dramatic prolongation or in median progression-free survival beyond what one might expect in the 3rd, 4th line setting. But what gave a little more hope was looking at the landmark analyses at 6 months PFS and 12 months PFS. And there was more of a hint, because a 6-month progression-free survival of 40% in a heavily pretreated patient population, that was notable. And 21% of patients alive and free of progression after a year, but that really also was quite remarkable. So this gave an indication that there was something happening with use of the targeted agent that perhaps not all patients were benefiting from the rapid decline here in the slope of this part of the graph, that there was perhaps a tail in that curve, that there were a significant population of patients who perhaps were benefiting. Again, looking at the response rate, well, this doesn't look like the waterfall plot that we might see with a typical, let's say, a tyrosine kinase inhibitor. So it doesn't look like that, does it? You can see that we had just 4 partial responses, some stable disease, quite a few patients with progression of disease. So again, a sense that we have a signal of activity, but hard to drill down on exactly where that signal is. And I think it becomes a little more evident when you look at the swimmer's plot. So again, I'm sure you're all most familiar, but those who aren't clinically based, each patient here represents a horizontal line, and the patients with stable disease are in green, with partial response are in blue, and with purple are those who progressed immediately. And this along the x-axis is treatment duration, so the length of time the patients were on treatment for. And obviously patients were permitted to remain on treatment for as long as they were benefiting from it. And what you can see when you look at this, thinking that every bar here is an individual patient, that there were some patients with really quite a dramatic benefit, right out at 2 years, continuing on treatment, continuing beyond mild disease progression, but with overall symptomatic benefit and clinical benefit. And then there was a clustering of the patients here who appeared to gain absolutely no benefit at all to the agent. Could that be that these were patients who unfortunately were too advanced in their disease process to benefit? Was it a dose intensity issue? But we looked at those and it didn't quite seem to be the case. It certainly seems to be that mutant IDH1, at least in the phase 1 study, was required but not sufficient for response to single agent targeted therapy. Having said that, it certainly was a signal in a disease with otherwise very little good treatment options, is particularly beyond conventional cytotoxic therapy in the 1st line and now 2nd line. And so a sort of brave move was made to move ahead to a phase 3 study. And, you know, it's easy to look at the schema and think, well, that's standard phase 3 study, looks good. I think it was actually quite ambitious to do a phase 3 study in a rare molecular subtype of a rare cancer. And so one important, I think, achievement of the Clarity study was that it was done at all, and that it accrued remarkably rapidly and almost ahead of expected timelines. And again, showing that it's possible to do these studies when they are done in a collaborative manner and with active patient engagement from the beginning. So this is a phase 3 study design. These are patients who'd had just 1 or 2 prior therapies, so not beyond 3rd line. They'd had gemcitabine or 5-FU. They were good performance status patients, centrally confirmed mutant IDH1 status, and randomization to placebo or to ivosidenib, that was a 2-to-1 randomization in favor of the experimental agent. And crossover was permitted at radiographic disease progression. So at time of disease progression, unblinding and crossover was permitted, and about 70 patients ended up crossing over on the study. So baseline characteristics, as one would expect, this patient population, most of them had had at least 1 or 2 prior lines of therapy. The IDH1 mutations were the most common ones that we usually identify in these patient populations. So just jumping to the overall survival analysis, this was presented by Andrew Zhu just very recently. And looking at the overall survival analysis, you see that it was just not meeting statistical significance, with overall survival of 10.3 months with ivosidenib versus 7.5 months with placebo. However, knowing that there was adjustment made for crossover, an additional statistical analysis performed to look at that, we did see that there was in fact a more significant benefit when one looked and adjusted for the patients who had crossed over. And looking at that sort of, again, landmark overall survival analysis, you do see that with even here, placebo versus ivosidenib, even not adjusted, you see a clear separation of the curves in that area. Progression-free survival, again, that primary endpoint was met in the study. There were very few responses on this study, so 2 partial responses, 2% partial responses, obviously none with the placebo. So again, seeing that pattern of disease control with stable disease, but not a lot of radiographic response seen. This efficacy was consistent across subgroups in terms of male, female, disease extent, the intrahepatic, extrahepatic subtypes, performance status, and geographic regional pre-planned analyses. So important to notice that in this study, 70% of patients crossed over to active treatment. So the adjusted survival analysis, which showed median overall survival of 10.3 months versus 5.1 months, did reach a statistical significance, p-value less than 0.01. There was also an improvement in health-related quality of life, and the study had already met its primary endpoint of progression-free survival. So a new drug application was submitted to the FDA just earlier this year, and anticipating perhaps an approval to come soon. And so again, emphasizing that this can be done in rare cancers, and a positive result for our patients to see that. But it doesn't stop there, because again, yes, with each incremental gain, we're pushing out that survival, but it's still not where we want to be. We're still looking to achieve better responses that will result in better clinical control of disease, and achieve less toxic regimens that maybe would be an alternative to chemotherapy in the first-line setting, in the perioperative setting. So how do we improve the outcomes for our patients while minimizing toxicity with these agents? And to think about that, we have to go back to the translational studies, because the positive part of the phase 1 in particular was the level of translational studies that were embedded within that, which still are producing a lot of interesting and useful data. And it's kind of humbling that the patients enrolled in that phase 1 study were extraordinarily generous, whereby they underwent research biopsies, so purely for research, of their disease at baseline, so pre-treatment, after 3 cycles of treatment, and after 7 cycles of treatment, and even some at time of disease progression. So that was an invaluable resource when joined with the blood-based markers, which has produced a wealth of information, or at least provocative information for further validation in future studies and in larger groups. So key questions to be addressed. We know that mutant IDH1 is required but not sufficient for response, so additional biomarkers are needed. What are the mechanisms of primary and of secondary acquired resistance to these agents? What is the role of co-mutations? Can we use mutational profiling to identify a particular subgroup of IDH1 mutant cholangiocarcinomas most likely to benefit from such an approach? And what's the optimal sequencing of these targeted agents with cytotoxics? Can we combine with immunotherapies, other targeted agents? And if so, how should we do that? And how should we sequence them? So the first question, co-mutations. Well, we looked at that association of co-mutations and multiple cancer-associated genes with both progression-free survival and response. These are patients going back to the phase 1 study, looking at baseline samples that had comprehensive molecular profiling performed. We didn't see a definite association or clear pattern in terms of genetic alterations that were associated with better clinical outcomes in these patients. So if there is a pattern to be found, it's not as simple as one that we can find from this exome sequencing. So here you can see they're all IDH1 mutant, obviously. Clustering up here, you have the patients who had stable disease, who had improved progression-free survival. Here, the patients who had upfront disease progression. Maybe a hint or a sense that the chromatin remodeling genes, perhaps that alterations in these may be associated with a better response to therapy, but nothing that actually was significant or that would fall out as a definite subgroup that should that clearly benefited. So then, to further drill down on that, we looked at the more extreme responders. So there were only a few patients that had responses to treatment. There were many that had stable disease and clinical improvement associated with that, but there weren't many patients who had actual radiographic response meeting RECIST 1.1 criteria. But in the few patients that did, the 4 that did, we did have a wealth of biopsy material and blood to deal with and to look at. So this is a patient who had a very good response to treatment. So here you can see liver tumor screening here, and you can see then by cycle 7, day 1, quite a significant response that did meet partial response by RECIST 1.1 criteria. And so we had from this patient a biopsy taken at screening at cycle 3, day 1, so after 2 months of treatment, and at cycle 7, day 1, so after 6 months of treatment. What we found is that there was overall gene expression changes that seemed to be associated with the patient with a partial response. And that seemed to correlate with increased expression of genes associated with hepatocyte differentiation. But then provocative, a small, just 1 patient, and we included this in the initial manuscript for the phase 1, just really to show that there may be a different mechanism of action of this drug in solid tumors that perhaps may mimic the mechanism of action of IDH1 alterations and targeting in the AML population, with the more differentiation effect perhaps seen. And that was mirrored also by looking at the morphological effects that are induced by treatment. Again, the same patient here, and a sense or a hint, at least the pathologists tell me, that there is a better differentiation and more organization of the biopsy by cycle 7 than one would have seen at the pre-treatment initial biopsy. So, hypothesis generating, but small numbers. So that led to a bigger study of patients enrolled in the phase 1 in the translational setting, which was just published last month. And this is a collaborative effort led by the company, but also by pathologists at MGH and MSKCC. And what they did here is they actually took the samples, which were matched pairs, baseline treatment and at cycle 3, day 1 of treatment, so after 2 months of treatment. And they did gene expression analysis, and they actually had 30 matched sample pairs, quite a lot for a phase 1, and again speaks to generosity of the patients involved and shows the importance and the legacy that these patients actually have provided by being able to contribute such important material to help us determine the mechanism of action and best next steps. So looked at genes associated with treatment, with response and with progression-free survival to see what's upregulated, what's downregulated in patients who did well and patients who did poorly. And what was identified, not just looking at the gene expression analysis, but also looking at the morphological analysis, was that there was a morphologic change in the biopsies following treatment with the IDH1 inhibitor. And what was seen was an increase in this sort of cholangiolar pattern and a decrease in cytoplasm following treatment with ivosidenib. This cholangiolar pattern is one that's typical of IDH mutant cholangiocarcinomas and tends to mimic sort of small duct cholangiocarcinoma type morphology. So here, just looking at the liver-specific genes associated with progression-free survival and response, you could see that there was induction of expression of liver-specific genes, but decreased expression of cholangiocyte lineage markers as well. And so you see here decrease in pancreatic keratin and EpCAM CK19. And looking at progression-free survival in the population, there was also a trend towards increased progression-free survival in those patients who had increased cholangio pattern and those who had cytoplasmic change as well. So essentially, from the initial analysis of phase 1, seeing that we did see IVSIDNA-induced morphologic changes and some evidence of hepatocyte differentiation, which did seem to correlate with some improvement or at least a trend to improvement in progression-free survival. So again, suggesting we may be looking at a different mechanism of action in solid tumor here, as compared to what we would normally see in a phase 1 study of a tyrosine kinase inhibitor, EGFR inhibitor, FGFR inhibitor. So there was evidence of hepatocyte differentiation with some decreased markers of biliary fate, cell cycle progression, stemness, and AKT pathway activity. But in patients, in fact, who had rapid progression of the study, there was an increase in AKT activity seen, more cell proliferation, more stem cell signatures. So suggesting that IDH1 inhibition may have induced differentiation of cholangiocarcinoma cells towards a hepatocyte-like phenotype in patients who had disease control. So acknowledging the sort of retrospective nature of all that and the relatively small numbers, albeit good for a phase 1 study, all this sort of hypothesis generating that requires validation in more prospective studies, but is thought-provoking in terms of how we might leverage that for additional benefit for our patients, and also how we might be able to utilize the initial biopsies and initial analysis towards perhaps identifying subgroups of patients who perhaps are more likely to benefit from treatment. So next question to address is that of acquired resistance to IDH1 inhibition. And this again was something that that was kind of difficult to elucidate, despite having the benefit of all these biopsies. But this was one resistance mechanism that became quite clear, at least in a minority of patients. So this study, we looked at a lot of patients in terms of molecular profiling pre, during, and post-progression of patients in the phase 1 study, not only from the cholangiocarcinoma cohort, but also from other studies, including AML and chondrosarcomas. But this is an example of a patient in particular with cholangiocarcinoma, who was in fact a really extreme responder on the study and had a very good clinical benefit with treatment, and eventually began to progress. So this was a patient who was over a year and a half on study. One of the first patients to go on study, had a lovely response and a rapid clinical improvement, and had a very sustained response to treatment. As you can see here, the initial large volume disease, excellent response after over a year on treatment, and then beginning of progression in this lesion here, rather than the main lesion. And this lesion was actually biopsied at time of disease progression. So what we saw was obviously this, initially looking at the RECIST response, a nice decrease, as we see here reflected in the tumor measurements. But the emergence in tandem with disease progression of this lesion was the emergence of a mutation in IDH2. So here, this is digital PCR for IDH1 mutation and IDH2 mutation in serum, so circulating tumor DNA analysis of the exact mutation that the patient had and of the resistance mutation. And so what we found is that in the biopsy taken here, there was continued presence of IDH1 R132C mutation, but the emergence now of an IDH2 R17V mutation. So pre-treatment, not present, IDH2 mutation, IDH1 mutation present at progression, IDH1 mutation present, and IDH2 mutation present. And so that correlated nicely with what we identified in circulating tumor DNA as well. And also, in fact, with measurements not shown here of 2-hydroxyglutarate in serum, which we were also able to measure. So interesting that this isoform switching was a clear mechanism of acquired resistance, that the tumor was so 2-HG dependent that it managed to acquire a mutation in the only other gene that could produce the oncometabolite in the same manner as the mutant IDH1. Now, so how do we overcome that strategy? Combined IDH1/2 inhibition, sequencing of those, and pulsing of those, but a nice example of isoform switching as that resistance mechanism. And in fact, when we go back to the phase 1 study data and some of the supplementary data, there also was another patient who had a stable disease for a reasonable long period of time, up to 19 cycles of treatment. So these are patients who remained on treatment for quite a long time. And we see here in the sequencing data of newly identified variants, this patient had initially an R132C mutation. And eventually, on biopsy at time of disease progression, R132C mutation was not identified. Instead, there was an IDH1 R132F mutation identified. And that's interesting because in vitro data, that is a mutation that is less sensitive to ivosidenib than the R132C. And in fact, by far and away the least sensitive of any IDH1 mutation to ivosidenib. So suggesting, well, did it actually switch to a variant which was actually resistant to the IDH1 inhibitor. So briefly looking forward, I suppose, where do we go to from here? And there has been, thankfully, a wealth of excellent preclinical data that has provided very good hypotheses as to where the next direction should be. Ongoing studies in many of these areas. So lovely work from Neil Badizi's lab looking at dasatinib, demonstrating initially that mutant IDH1 cholangiocarcinoma is highly dependent on SERC for survival, for protection. That dasatinib, which inhibits SERC as a multi-tyrosine kinase inhibitor, is quite effective in vitro, at least, and in vivo, and ongoing studies of that. We mentioned earlier on about the effect of IDH1 mutations on DNA repair. And it does appear in vitro, at least, that 2-hydroxyglutarate may enhance sensitivity to PARP inhibitors in vitro. And so again, ongoing studies targeting the DNA repair pathway in patients. Combinations with immuno-oncology drugs, as we are now mandated to do in every study, but in fact there is a rationale here. And again, in samples from the phase 1 and then additional preclinical data from other laboratories, it has been shown that IDH1 inhibition can increase PD-L1 expression. And so a strong rationale for that combination in this patient population. And then the obvious combinations with first-line chemotherapy, perioperative treatment, neoadjuvant, adjuvant treatment. Should it be sequenced? How do we do that? Does it work in the adjuvant setting? All these good questions, clinical questions to be addressed. And then obviously the newer IDH1 inhibitors, in particular FT2102, which has actually very, very good brain penetration. So just to overview ongoing studies, most of which we've mentioned, novel inhibitors. A PARP inhibitor actually being developed with an ATR inhibitor, which is a nice strategy. The ongoing study, cisgem in combination with ivosidenib or pemigatinib, depending on the molecular profile of the tumor type. So again, really encouraging to see the level of activity, the number of studies available to our patients. Of course, we'd love to double it and have multiple lines of studies for patients, but really, I think, encouraging to see that over the last decade, when we had really a dearth of studies for these patients, that now we have not quite a wealth, but certainly a reasonable amount. So concluding then, success, yes, we have some success in targeting mutant IDH1 cholangiocarcinoma. Ivosidenib was the first targeted agent to show benefit. But that's just step one. There's still much to do, and we're really not where we need to be in terms of significantly better outcomes for our patients. So addressing from a translational perspective, key questions, need to identify and address primary and secondary resistance mechanisms, need to identify markers of sensitivity to IDH1 inhibition and ways to alter that with drug combinations. Multinational collaborative efforts are essential, like this, joining people together when this kind of a rare tumor requires international collaborative efforts to understand the disease better, to provide options for our patients no matter where they are, and to learn from each other in a narrow field. And possibly that involves these sort of next generation studies. Maybe we need to look at adaptive platform designs, of course, embedding translational endpoints. And again, not listed here, but emphasizing the importance of patient advocacy and excellent patient advocacy organizations, combining academia and industry involvement, leveraging that directly for patient benefit, and embedding patient involvement from the very, very beginning. And that's something that I know the Cholangiocarcinoma Foundation have done a wonderful job at doing, and is in large part responsible for the successes that we've seen over the last few years. So I'll leave it there. I'm delighted to take any questions from the audience. And oh, here's a nice picture of where I work. This is Trinity College at sunset. Not in the rain.
Thank you so much for that tour de force, and frankly for your efforts over the last few years to show us what we can do in biomarker directed studies in our cancer type. I just think it's been such an amazing few years for us, both with your work and what's been done with FGFR as well. I would love to hear your thoughts on why what we're seeing with FGFR mutated or fused tumors is different than what we're seeing with IDH1, and biologically why you think that there's such a difference between the amount of benefit that we're seeing, or do you think there's a difference?
Yeah, I think it's really different. I always think of the FGFR inhibitor sort of platforms, if you like, more like targeting EGFR. You know, the more similar thing of what we'd expect to see when we have, you know, targeting a single receptor, where you get that emergence of resistance with gatekeeper mutations around about the 4-6 month mark, that you have quite a bit of toxicity maybe with the drug or particular off-target effects from the drugs. I think they fit more neatly in the drug development paradigm we've seen in lung and, you know, BRAF inhibition, EGFR inhibition, not quite HER2 inhibition, but that to me is more the conventional, if you like, pathway, where you don't get quite so much durable responses, but you get much quicker and deeper responses early. And that was seen even in the early studies of the FGFR inhibitors, even with the Novartis compound. That was when the first studies, patients responded deeply and quickly, but then almost universally progressed around the 6-month mark. And we didn't see that in this phase 1 of the IDH1 inhibitor. We saw, at least what I saw in my patients, which was something I hadn't seen before, was clinical benefit without radiographic response. And that to me was unusual, because, you know, when we treat these patients, Nilo, you almost always have to get a response in a GI cancer patient to make them feel better. You know, it's quite rare to have a patient have pain resolved and feel better and put on weight without having radiographic response. That's really unusual. And I think that's really fascinating. And that's why I think this translational aspect to the program is essential, because it really is quite unusual. I do think we're seeing a different effect in terms of the actual cytotoxicity of the drug as compared to the usual TKI paradigm.
Yeah, I totally agree with you. I'd also like to invite any of the people that are on the call right now to put any questions into the chat box as well. But I treated a patient who was on for 1.5 years on the phase 1 study, and she had pleural effusions, felt so much better, tolerated the drug so well, didn't have a single response, you know, and even a couple of times looked like she might have a tiny bit of progression, but then that would even stabilize, but really, you know, did beautifully for 1.5 years, which you never see, right? I have another question for you. So you had a very nice slide of the potential future options for IDH1 mutated cancers, but as someone who's very interested in epigenetic therapy, I'm just curious what you think about the role of potential epigenetic modulators in an abnormality that really results in widespread epigenetic dysregulation.
Yeah, totally. I mean, I totally agree. I think that's exciting. But I think we have work to do, and it sounds almost crazy to further subtype a rare subtype of a rare tumor, doesn't it? But I have a feeling that we need to better characterize the epigenetic status or alterations within that. I think that might change. I think that might change over with treatments, so with cytotoxic therapy, maybe with targeted therapies. So I'd love to see us better delineate that biomarker, if you like, with sequential biopsies or sequential patient treatments, if you like. Because I think it's kind of a dynamic situation. What struck me this patient population is that there were clear patients who had mutant IDH1 that had no benefit at all from the drug, you know. Something really different about those patients. And maybe it is the extent of epigenetic dysregulation. Maybe it's the methylation status. That didn't seem to correlate on the initial translational studies, but there's something different about certain patients there. So I think we have to move away from assuming that mutant IDH1 cancers, cholangiocarcinomas, act in a uniform manner, and more look to see that there may be different effects of mutant IDH1 at different time points in the patient journey. It just affects so much parts of the key cellular mechanism that it's just, there must be different parts that are prioritized at different times.
Absolutely. There's a question in the chat box. Have you seen patients that have benefited from therapy post-progression on AG120? And what are the potential immune effects that are possible with both the inhibitor and with the biology itself?
So first question, I'm always wary of treatment beyond progression, but I did do it a couple of times in this study, and it was really for those patients, a bit like the patient that I showed you with the IDH2 mutation in that one solitary lesion, is that what I found a lot of patients who were on study for quite a while had maybe just one area of disease progression, but all the others remained totally without progression. And so you were allowed to sort of do a local therapy, radiate a small bone lesion that was growing. And so I think, again, for these patients who were a long time on study post-progression, it was asymptomatic progression, small volume, and usually one site only, with disease controlled at all other sites. So so I don't think it's the same paradigm as the immuno-oncology beyond progression. This is really rather, there was definitely an escaped lesion, be that to whatever secondary mechanism of resistance that it had acquired, but there was definite clinical benefit to continuation of the treatment. So I think yes, but only in a subgroup of select patients with clinical stability and a small volume or isolated area of progression. If they were progressing at all sites, I had no patient that benefited from that. The second question about combination of immuno-oncology agents, I mean, I think that's really exciting. So again, I didn't show you the data again from the phase 1, but there was analysis done looking at sort of PD-L1 expression in patients who had cycle 3 and a cycle 7 and post-treatment biopsies. There was an increase seen in PD-L1 expression there. There's other papers that have shown a similar effect. And I like that there's almost no overlapping toxicities. So it appeared to be, we'll see from the data from the studies, but it would appear to be a nice, safe combination for patients and one that might provide good quality of life as well as potentially good response. But I guess we'll wait to see the outcome of that study.
I see another question asking about the combination of IDH1 inhibition and hypomethylating agents, which has been tested and is in phase 3 in liquid tumors, and what you think about that strategy for cholangiocarcinoma.
I think it's, I guess we don't know until we try. I think that the IDH1 mutation, I think, is very different in liquid and solid tumors. So really different, and the level of responses and efficacy that we're seeing are very, very different. So we can't assume that there's going to be the same effect. I think drug delivery is an issue, that there isn't an issue in the liquid tumors. So I mean, yes, but again, I think it's all, again, that would lend itself, this small patient population to that, in my last slide, that kind of idea of an adaptive platform design. You can have a good idea testing a small number of patients, and if it looks promising, then you move rapidly forward. It was kind of a brave move with the ivosidenib to go from phase 1 to phase 3, but mightn't have worked. See, the idea of being able to roll out in and out different arms, then patients to move to different arms of a study as they progress, I think maximizes potential for patient benefit and maximize our ability to rapidly test these kind of combinations with good, strong preclinical rationale.
Yeah, and I think it allows us to not have to go through the entire exercise of designing and opening a new protocol every time you want to tweak what you're going to give a patient. So yeah. I am hopeful those kind of platform studies will be able to get underway. Well, thank you so much. I think this has been a fantastic session, and we're going to look forward to being able to see all of each other in person sometime in the next year. But I'm going to wish everyone a wonderful weekend. I'm going to thank you again for being with us today.
Yeah, thanks so much, everybody.
Bye.